Good day, everyone. Before we dive into the specifics of transcription and translation, let us take a step back and understand the bigger picture. At the heart of every living organism lies a remarkable set of molecules, nucleic acids, that carry the instructions for life. Next slide, please. Today, we'll walk you through this entire journey. First, starting with an overview of the genetic blueprint, then exploring the processes of transcription and translation, then followed by how these steps lead to the conversion of genetic code into functional proteins. And lastly, we'll wrap up by discussing some notable exceptions to the central dogma that challenges and expand our understanding. Next slide, please. Now, let us begin our exploration of how life expresses its genetic code.
The genetic blueprint focuses on the DNA, the molecule that holds all the genetic instructions necessary for life. This part plays a central role in the central dogma of molecular biology because it serves as a starting point in the flow of genetic information. Next slide, please. But what is a genetic blueprint? A genetic blueprint is essentially the complete set of instructions encoded in an organism's DNA. Think of it like an architectural plan for a building and maintaining all the structures and functions of a living organism. DNA stores this blueprint in the form of specific sequences, which eventually guide the synthesis of proteins through transcription and translation. Next slide, please. Now let's talk about structure. DNA or deoxyribonucleic acid is made up of repeating units called nucleotides. Each nucleotide contains a phosphate group, a deoxyribose sugar, and one of the four nitrogenous bases adenine, thymine, cytosine, and guanine. These nucleotides link together to form double helix. The strands run in the opposite directions, what we call anti-parallel, and the bases pair up specifically. Adenine pairs with thymine, and cytosine pairs with guanine, held together by hydrogen bonds. Next slide, please. DNA has three major roles. First is the storage of genetic information. DNA contains genes, which are specific sequences that codes for protein. Second is the replication. DNA can copy itself during cell division, ensuring genetic information is passed down accurately. Third is stability. Its structure makes it chemically stable, which is critical for storing information. During replication, enzymes like DNA polymerase, helicase, and ligase help unwind, copy, and seal the DNA strands. This replication process is semi-conservative, meaning each new DNA molecule has one old and one new strand. Next slide, please. Organization of DNA. In prokaryotes, DNA is typically circular and found in the cytoplasm. In contrast, eukaryotic DNA is linear and stored inside the nucleus, wrapped around proteins called histones to form chromatin. Genes are segments of DNA that code for proteins, while non-coding regions play important roles in regulation. Next slide, please. So how does DNA fit into the central dogma? DNA serves as the template for transcription where it is converted into RNA, which is then translated into proteins. The sequence of bases in DNA ultimately determines the amino acid sequence of proteins. Sometimes mutations like point mutation or deletions can alter these sequences, which impact how proteins are made or function. Some are harmless while others can cause diseases.
Now, why don't we take a closer look to the transcription, which is a process of synthesizing RNA from DNA. Transcription consists of three stages, which are the initiation step, the elongation step, and the termination step. So first, for the initiation step, next slide please. The initiation step starts when all the necessary pre-initiation complex proteins assembles around the code promoter of the gene. So this complex composes of RNA polymerases and transcription factors. The transcription factors are proteins that combine to the DNA that regulate gene expression. So to start the transcription, RNA polymerase requires several different proteins called the general transcription factors to bind to the promoter regions. These transcription factors are composed of TF2D, which is the largest, while the others are TF2B, TF2E, TF2F, TF2H. And next slide, please. Then TF2 binds to the Tata box found in the promoter, and once binded, the other proteins will also bind and recruits the RNA polymerase, forming the pre-initiation complex or PIC. Yeah, next slide. And in eukaryotes, there are three RNA polymerases. So the first one is RNA1, which transcribes ribosomal RNA genes, and RNA3, which transcribes tRNA and snRNA genes. And for RNA2, it transcribes the majority of the protein encoding genes. Next slide, please. Then after this step, the RNA polymerase unwinds a short stretch of DNA upstream of the transcription site. And once the initiation stage started, the transcription factors were released and the elongation begins. So RNA polymerase will begin to produce a new strand of mRNA using the template strand as the guide. Nucleotides are added one by the RNA polymerase using the base pair rule. And this occurs at 5' to 3' direction and produces a copy of the coding strand. However, the thymine will be replaced with uracil. And this process will continue until the termination sequence will be encountered that will release the newly made mRNA that will undergo further processing. So next slide, please. Shown in the slide is the summary of the transcription. Next slide, please. So after we synthesize pre-mRNA, it will undergo two important modifications, which is the first, the 5' capping and poly A tail, and the next one is RNA splicing. So the first modification is 5' capping, or this is where first is the 5' capping, where a single methyl guanosine attaches to the first nucleotide of pre-mRNA via a triphosphate linkage. And next slide, please. And the second step is the poly A tail at the 3'. So in this modification, a polyadenylation signal nucleotides, usually as shown in the slide, AAU, AAA, recruits a RNA binding protein which guides the endonucleases to cut the transcript at the 3' end of the signal sequence. Then a different enzyme called polyadenylate polymerase adds a long string of adenine nucleotides, as many as 200, to the 3' end of the transcript. The next one is RNA splicing. So in this process is where proteins complexes called spliceosomes remove introns or the non-coding sequence of RNA. And they connect the exons or the coding sequence of RNA to each other to finally produce the mature mRNA that is ready to leave the nucleus to the nuclear membrane called nuclear pore complex.
Molecular symphony of life, central dogma of molecular biology plays a defining role. stating how genetic information flows from DNA to RNA to protein. In this intricate journey, translation stands as a pivotal process, the phase where the nucleic acid language of RNA is translated into the amino acid language of proteins. It is through translation that the genetic instructions encoded in DNA ultimately manifest as enzymes, receptors, hormones, antibodies, or structural protein, which are the molecular machinery that sustains our life. With that in mind, let us now examine in detail the molecular logic, the key players, and the stepwise mechanisms of translation. But before diving into the mechanics of this process, it is essential to introduce the fundamental molecular entities involved in translation. First of that is mRNA or messenger RNA, which contains the codon sequence that determines the order of amino acids in the protein. Each group, each group of three nucleotides forms a codon that corresponds to a specific amino acids or a termination signal. Next one is transfer RNA or tRNA. This is the folded RNA molecule that carries a specific amino acid and possesses an anticodon region complementary to the mRNA codons. This serves as the adapter molecule that interprets the genetic code. Next one, we have ribosomes. Ribosomes consist of two subunits, the small subunit, which decodes the mRNA, and the large subunit, which form peptide bonds. This features three binding sites for tRNA, which is the A site or aminoacyl site, which is the entry point of the charged tRNA, the P site or peptidyl site, which holds the tRNA with a growing peptide chain, and lastly, the E site or the exit site, which is the site from which empty tRNA exits. Next one, we have amino acids and peptidyl transferase. Building blocks of proteins joined by peptide bonds catalyzed by peptidyl transferase is a ribosome activity of the large subunit. Lastly, we have the GTP and translation factors. These are the energy molecules and protein factors that aids in the initiation, elongation, and the termination process that happens during the translation. Next slide, please. Okay, thank you. Translation occurs in the cytoplasm of both prokaryotic and eukaryotic cells. In prokaryotes, it begins even as mRNA is still being synthesized, which is a phenomenon known as coupled transcription and translation. In contrast, eukaryotes undergo translation in the cytoplasm after post-transcriptional modifications, such as 5' capping, polyadenylation, and splicing have refined the primary RNA transcript into a mature mRNA. Central to translation is the ribosome, a dynamic ribonuclear protein complex that is composed of a large and small subunit. These subunits, differing slightly between prokaryotes and eukaryotes, converge at the mRNA strand to catalyze protein synthesis. This process begins when the small subunit of a ribosome in a complex with a methionine tRNA and initiation factors binds to the 5' end of an mRNA. The complex scans the mRNA in the 5' to 3' direction until it encounters an AUG codon that serves as a translation initiation site. Next slide, please. Initiation is assembly phase, wherein the ribosome complex scans the mRNA and identifies the start codon, positioning itself correctly on the mRNA. In eukaryotes, the small ribosomal unit or subunit binds to the 5' cap of the mRNA and scans in the 5' to 3' direction until it encounters the start codon, which is AUG. Next slide, please. Here in this slide shows the codon chart, which is a fundamental tool that is used to decode the genetic information carried by mRNA during translation. It maps all 64 possible tree-based combinations of RNA nucleotides or codons to their corresponding amino acids. To read the chart, you match the first base from the left column, the second from the top row, and the third from the side column to find the correct amino acid. For example, the codon AUG codes for methionine, which also serves as the universal start codon. Of the 64 codons, 61 code for amino acids, while 3 of them acts as stop signal to terminate protein synthesis, which are UAA, UAG, and UGA. The genetic code is also redundant, meaning multiple codons can specify the same amino acid, a feature that provides a buffer against some mutation. This is due to the wobble position in the third base, wherein variations often don't change the amino acid being coded. During translation, each codon is matched by a complementary anticodon on a tRNA molecule that carries the correct amino acid as defined by the codon chart. This systematic pairing ensures the ribosome builds a precise and functional protein from the mRNA sequence. Next slide, please. AUG codes for methionine, and this codon is universally recognized as the beginning of translation. Next of that is that a charged tRNA bearing methionine, or the initiator tRNA, pairs with the start codon via its anticodon. Next slide, please. After that, a large ribosomal subunit then joins the complex, completing the assembly of the functional ribosome. After this, the ribosome is now ready to build a protein, one amino acid at a time. Next, we move to the process of elongation, which is a cyclical and processive phase where amino acids are sequentially added to the growing polypeptide chain. Next slide, please. Okay, so first of that is codon recognition, wherein a charged tRNA enters the A site, or amino acyl site. Its anticodons matching the codons currently exposed on the mRNA. This ensures specificity and fidelity in the amino acid sequence. The second is peptide bond formation, wherein the ribosome catalyzes a peptide bond between the amino acid attached to the tRNA in the P site or the peptidyl site and the one newly arrived in the amino acyl site. This reaction is mediated by peptidyl transferase, a ribozyme function on the rRNA within the large subunit. The next process is the translocation, which is the critical shift. In this process, the ribosome must now reposition itself to read the next codon, which is called the translocation process, as I've said earlier. This is processed by the GTP hydrolysis, wherein the ribosome shifts forward by one codon along mRNA strand. The tRNA that was in the P site, which is now uncharged, moves to the E site or the exit sites and exits the ribosomes. The next of that is that the tRNA that was in the A site or the amino acyl site, now carrying the elongating polypeptide, shifts into the peptidyl site. The amino acyl site becomes vacant, exposing the next codon, and this coordinated movement involving large conformational changes in the ribosome occurs with every amino acid addition, repeating over dozens and even hundreds of codons. This cycle ensures that the genetic code is translated into a linear amino acid sequence with extraordinary accuracy.
Once the mRNA is translated, the ribosome has already done its part. So in order to reset, it needs to be dissociated from the RNA to completely produce newly and ready formed peptide and polypeptide chains. But when does it start? It begins when the prolongation or elongation process of tRNA no longer matches the specified codons in the mRNA. These codons are known as the stop codons. Stop codons typically varies among three specific types of codon. We have the UAA, UAG, and UGA. When these codons reach the mRNA, the anticodon of the tRNA that is supposed to enter the A site have been inhibited due to unmatching of the bind with the stop codons. Hence, no binding will be done. However, that doesn't mean that the process ends there. The presence of stop codons are recognized by a special type of proteins, and these are called the release factors. Release factors have its role. The primary role of RF, or the release factor, is to bind with stop codons and promotes the release of polypeptides. RF1, which are used for stop codons such as the UAA and UAG, and RF2 is for those UAA and UGA codons with the currently presented stop codons. And once it binds, it forces the enzyme peptidyl transferase in the ribosome to catalyze the addition of water molecule instead of the amino acid to the peptidyl tRNA. With this process, the water molecule breaks the bond between With this process, the water molecule breaks the bond between the polypeptide and tRNA in the P site, releasing the newly made polypeptide into the cytoplasm. After the release of the polypeptides, a third release factor, specifically RF3, bound to GDP, joins the ribosome after the RF1 and RF2 takes place, leading for it to bind with RF1 and RF1 or RF2. While on ribosome, RF3 replaces the GDP with GTP. This happens automatically as the ribosome promotes this exchange. So the switch of GTP to GDP and binding with the RF3 phenomenon causes a conformational change in the ribosome. As rearrangement of the ribosome causes non-rotated state, which is used typically for decoding and peptide bond formation, to a rotated state. This change affects the subunit interface, which leads to open up the ribosome structure. The rotated ribosome disrupts the binding site of RF2, or also it can be the other way around, in where the RF2 in the A site destabilizing, causing them to dissociate. So after the dissociation, RF3 catalyzes GTP hydrolysis, forcing to undergo conformational change in RF3 itself back to its inactive form, acting like the switch or lever. And this will eventually allow subunit to dissociate from each other and the mRNA. So after disassembling, the ribosomal units bound to an initiator tRNA and can now repeat the process again back to initiation step. And the whole process will be repeated. So in this slide, this is a summation of the translation process, highlighting the initiation step in where the ribosome, mRNA, and signal peptide are involved. We also have the prolongation and the elongation step, which involves the SRP receptor protein and also the translocation complex that are being discussed earlier. And lastly, it also involves the termination step in where the signal peptide is removed and successfully transfer or let go the protein. So just to give you a brief discussion on what would happen to the polypeptides after the central dogma, let us discuss. So after the release of polypeptide chain from the ribosome, it undergoes a series folding and may receive post-traditional modification to become a functional protein. So this protein folding, this protein folding typically undergoes 4 major steps. So we have the primary, secondary, tertiary, and the quaternary structure. So first is the primary structure, or also known as the starting point of protein folding. The primary structure is the sequence of amino acids in a protein chain. These amino acids are linked one by one by a peptide bonds. You can differentiate it like a beads on a string. This sequence is determined by the gene that codes for the protein, and each amino acid has its own properties. And some of these are charged, some are hydrophobic, and others are polar. And these properties individually influence how the protein will fold later on. So next one is the secondary structure, the local folding patterns. So for the secondary structure, once the amino acid is formed, or once the amino acid chain is formed, parts of it start to fold into specific patterns. These are called the secondary structures. This structure involves 2 major variables. We have the alpha helix and the beta pleated sheets. So for alpha helix, it's like the spiral shape, which looks like a spring, and the beta pleated sheet is like the zigzag shape. Okay, so how these shapes are formed? These shapes are formed by hydrogen bonds between nearby amino acids in the backbone of the chain, which helps to stabilize the structure and give it strength. These folded sections are just like the primary protein structure are important as it represent as the building blocks of the whole protein. They help create a more organized structure and often appear in the important areas of the protein like the binding sites or the active sites. Next is we have the tertiary structure or the final 3D shape of a single chain. The tertiary structure is when the entire protein chain folds into its final 3D shape, and this happened because of the interaction between the side chains or the R groups of the amino acids and not just the backbone. So these interactions include four important variables. We have the hydrophobic interactions, which happens when the nonpolar side chains hide from water inside the protein. And we also have the hydrogen bonds, which are formed between polar side chains. Next is the ionic bonds, which are made by the bonding between positively and negatively charged side chains. And lastly, we have the disulfide bridges, which is a strong covalent bonds between sulfur atoms and the cysteine residues. This level of structure gives the protein its unique shape and function. For example, enzymes have a specific 3D shapes so they can bind together by their target molecule. It's a protein misfolds at this level, if a protein misfolds at this level, it can lead to a certain diseases like Alzheimer's or Parkinson's, where misfolded proteins forms a clump in the brain. Okay. So lastly, we have the quaternary structure or the multiple chains working together or the combination of the tertiary structures. So when two or more folded subunit chains or like the tertiary subunits or structure come together, it is called as the, just like I said, the quaternary structure. So in this structure, the subunits are held together by the same types of interactions found in tertiary structure, such as hydrogen bonds and hydrophobic forces, which makes the structure as important as others. And why is that? It is important because majority of the proteins only become functional when all their subunits are connected and working as a team. This level allows proteins to change shape when doing their job, which can be important and crucial factor in processes like muscle contraction or immune response. One good well-known example of quaternary structure is the hemoglobin, which is made of 4 subunits and is responsible for carrying oxygen in red blood cells and transfer to the whole body. And that is a summary of the conversion to proteins.
So, for the exceptions to central dogma, let's kick off our review by talking about reverse transcription, a classic exception to the central dogma. Normally, we think of genetic information flowing from DNA to RNA to protein. But in some viruses, especially retroviruses like HIV, the flow goes backwards. These viruses use an enzyme called reverse transcriptase to convert their RNA genome into DNA, which then integrates into the host genome. This means the cell starts transcribing and translating viral genes as if they were its own. This reversal of information flow, RNA back to DNA, is a powerful reminder that biology often makes exception to its own rules. Next slide, please. Next up is RNA replication, another viral twist. While our cells don't normally replicate RNA from an RNA, many RNA viruses do exactly that. Instead of going through a DNA stage, they copy their RNA genomes directly using a special enzyme called RNA-dependent RNA polymerase. This is crucial for viruses like influenza and SARS-CoV-2, which must quickly replicate large amounts of RNA to hijack the host machinery and spread rapidly. So in this case, we see an RNA to RNA pathway completely bypassing DNA. Next slide, please. Moving on to non-coding RNAs. These are RNA molecules that never get translated into proteins, but don't let that fool you into thinking they're unimportant. In fact, they're they're essential. tRNAs bring amino acids to the ribosomes. rRNAs form the core of the ribosomal structure, and regulatory RNAs like miRNAs and siRNAs help control gene expression by silencing or degrading mRNAs. So even though they don't make proteins, non-coding RNAs are critical for making sure the cell runs smoothly. Now let's talk next slide please. Now let's talk about prions, one of the weirdest exceptions to the central dogma. These are infectious proteins that don't carry any nucleic acids, no DNA, no RNA. Prions are cause diseases by misfolding, then include inducing normal versions of the same protein to misfold as well. This leads to diseases like Creutzfeldt-Jakob disease in humans, or mad cow disease in cattle. It is a radical conception, information being passed from one protein to another without involving any genes at all. Next slide please. Then we have RNA editing, which adds another layer of complexity to gene expression. After an mRNA is transcribed from DNA, it can be chemically altered. Nucleotides can be inserted, deleted, or changed. This means the protein produced might be different from what the DNA originally coded. A good example is apolipoprotein B, or ApoB, in humans, where editing of the mRNA results in two different protein products from the same gene depending on the issue. Next slide please. Lastly, we can't forget epigenetic changes. These are changes in gene expression that don't alter the DNA sequence itself. Instead, they involve chemical modifications like DNA methylation or histone acetylation, which control how tightly DNA is packed and how accessible it is to the transcription machinery. Epigenetics plays a huge role in development, self-differentiation, and even diseases like cancer. Two identical twins, for example, can have different epigenetic profiles and thus express genes differently over time, despite having identical DNA. Next slide, please. To wrap up our presentation, let's reflect on the central dogma as a whole, not just a diagram or flowchart. It's the central framework that helps us understand how our genetic code is interpreted into action. DNA holds the instructions, RNA acts as the messenger, and proteins carry out the function. This linear flow allows what life to sustain itself, reproduce, and adapt. Next slide, please. But as we explored, there are fascinating exceptions. Reverse transcription, RNA editing, and epigenetic changes all challenge the simplicity of the dogma and remind us that biology isn't rigid, it's dynamic. These exceptions don't invalidate the central dogma. Instead, they expand its relevance in the evolving landscape of molecular biology. In closing, the central dogma remains a cornerstone of life sciences, and understanding it, along with its exceptions, gives us a more complete and powerful view of how life operates at the molecular level. Next slide. Tapos na pala. Next slide. So that is all, and thank you for listening to our video.